All right, we're going to do a little bit of machine learning. I'm going to open my file in my Google Colab directory on my own drive. And what our goal is going to be is to see if we can create some machine learning algorithms that can tell whether a patient has either cancer or not. Let me start with some slides here. So what this actually, these two pictures are, before we kind of get into it, is that imagine that someone has a lump in their breast and you want to decide whether that lump is cancerous or not. Well, one way we do that is by taking a biopsy and then putting it under a slide and taking a look at it. So these are two biopsies where you basically take a small bit of tissue, put it underneath the microscope, stain it, and then decide whether it's benign or malignant. And this is what pathologists do. Now, I'm not a pathologist, but they have learned many different techniques on how to decide whether this is benign or malignant. What we're going to try to do is see if we can take some features that are automatically recognized from this by a computer and see if the computer by itself can recognize whether this slide is benign or malignant. And as you can tell right away, there's some differences that are there. Just to get a little bit of an overview of what the differences are, this is sort of a general overview, just to get a sense. It'll actually probably hopefully help you understand this lab a little bit better by having just a little bit of understanding. And for a normal cell, there'll be a lot of cytoplasm versus the nucleus. So large cytoplasm, single nucleus, single nucleoles, which are these here, and then very fine chromatin. Whereas in this side, whereas cancerous, we're thinking is going to be coarse chromatin, there'll be multiple and large nucleoli, and also could be multiple nuclei. And as you can see, you're kind of bumping up with less cytoplasm. So those are kind of the features that a pathologist would use to decide which of these slides are either malignant or benign. Fortunately, you can actually get a data set from online. If you just click on this link, you'll find there's a data set from Wisconsin that we can use, and it's just a text file. So if you click on, you'll be able to download that text file. Make sure you put that text file into the same directory as your Python code that you're running your Google Colab. And I'll put links to my Google Colab so you can actually download it directly and do it on your own. We're going to use a number of different classifiers, machine learning algorithms, just kind of get a sense for how they operate and just also the words that you might hear online. So we're going to do a standard MLP classifier, which is just a standard neural network using scikit-learn, which is very popular. We also use logistic regression, random forest, k-nearest neighbors, and support vector machines. Don't be concerned if you don't understand all these different terms. At this point, we're just trying to use it and play with it. And just imagine it's a system where you can actually put in numbers and then get out a prediction. That's all that it really is. It's just an algorithm. And if you want to delve deeper into that, you can definitely take courses that will allow you to learn a lot more about these different types of classifiers. Here, we just want to practically use them and get a sense what they feel like. We should have already downloaded the data above, but just remember that you can actually go through Kaggle, and there's a bunch of places online to get a lot of data. So I think Kaggle is one of the best places you can go. So definitely, if you don't have a account, I would go there and get an account to use that. Now, if you click on the link above, you'll download a CSV file. If you upload that file directly into your Colab directory, here's my Colab directory. This is my file. This is the data.csv. Suppose I can click on it right here. Let's see what happens when we see this. Oh, that's great. So you can kind of see. So there's a bunch of columns with a bunch of, these would be called features. This is the mean radius. This is the texture, as we talked about. But the average texture, a computer is actually computing this automatically. There's just an image analyzer that looks at all of these. Let me go back to that thing here. It'll take a slide, look at this image, and calculate the average mean radius. And it also calculate the average mean texture of all these cells. And then hopefully with having all these numbers, if we teach an algorithm, hold on a second. If we teach an algorithm, if we give an algorithm all these numbers, it'll be able to predict whether something is M, which is malignant, or B is benign, based on all these features. And as you can tell, that when you actually look at the slides, you can tell there's got to be some difference. And so the goal is, can we create a computer algorithm that does that? And the key thing to remember is that all these numbers are actually automatically calculated by a computer imaging algorithm. So all this is computer rated in terms of the collection of the features, and the only thing that is input here is the actual diagnosis that has to be done by, I believe, by a pathologist. Although they don't have the exact information. I assume this was done by a pathologist looking at the slides and deciding which are benign or malignant. So once again, as a reminder again, that this is going to be larger nuclei and maybe multiple nuclei, and that's how you're going to tell the difference between, and larger cells in general compared to the more normal cells. And you'll see that if you look at our data set, which we will load up, let's see if we load it. So we've got the pandas loaded. In order to load it off of Google Colab, you click here. This actually will mount the drive directly. So if you look at all your files, you can actually look at your drive. This is my drive. I believe I have it in here. And here's my data CSV. So when I run this code here, it will load up that file. This is the path to that file, and you should be able to use your own path for your own file and be able to get that data. So that's the first step, is always getting the data. So if you click here, just by typing df, which is, remember, that's our file that contains the data. Essentially, it's a matrix, and that matrix has 569 rows and 33 columns. These columns would be considered the features, and otherwise you would call this a target variable. In this case, we're just calling it the diagnosis, but this might be your target. These are all your features. There's a lot of features. Kind of scroll all the way across. There are 33 columns, or 32 features. Oh, actually, well, 30 odd features that are there. We're going to try to clean up the data, so we'll do that next. Now, if you type df, this is just a little bit of learning about what the different things you can look at df, make it a little bit easier to look at. If you do df.head, you can look at just the top of the file. If you do df.tail, you can look at the bottom of the file. You can put in here a number like 20, and then you can just get maybe 20, as opposed to just the default, which I believe is a lot smaller. We can count that. If you don't put anything in there, it's a default. Looks like you get 1, 2, 3, 4, 5. Okay, so it gives you a sense of what's going on. You can see these numbers are, they vary. They're like, 10s and 20s. Some of these numbers are in the thousands. These are a little bit smaller in the 0.1s, and that'll be important as we kind of look later, because that will actually mess up your machine learning algorithm if all these numbers are of different scale. But we'll fix that. In order to get a kind of visualization of what's going on, let's look at just one single variable. So I'll run this code here, and we're going to look at just one of these, radius mean, and we're going to look at the difference between the ones that are benign and ones that are malignant. It's really important to actually visualize all your data, and you can go through actually looking at each of the different features, but we're just going to do just one for right now, which is just the radius, to give you a good sense for what's going on. And I think your intuition right away, if you look back up here, the intuition is that these cells are a little bit bigger and these are a little bit smaller. So radius should hopefully give us some sort of idea of what's going on. And you can see right here that the malignant cells, yes, they tend to look like they're probably going to have a higher average over here, versus the benign ones are going to be a little bit more narrow, and also gives you a sense that there's not exactly the same, but it's not as if the number of malignant cells are very tiny compared to the number of benign cells. That's important. You want to have a balanced data set. We won't talk about that too much here, but this is a relatively well-balanced data set, and that means that there's a significant number of both malignant cells and benign cells, and there's a lopsided distribution. We can use the describe function to look at the actual mean, and this actually of all of these, and the mean you look is probably going to be somewhere around here. The calculation is 14, so that's where the mean is. A little bit more cells are down here, which moves the mean down a little bit further. Okay, we're looking at both the malignant cells and this mean is the malignant cells and the benign cells. And you can look at the average rate is about 13.7. Okay, and you can the mode as well. You can get all the different statistics you want, and you can look under. Actually, I would just use I use Gemini. You can use any large language model to learn anything about all these different variables, both radius mean mode or radius mean. You can also run describe on the entire data file. And it kind of, it doesn't really tell you much about radius. You can see we're back to 14 for the mean. Texture is around 19, average, and then we have a bunch of numbers here. So the first step is, that's a lot of data. So we're first going to need to kind of clean up the data. Let's kind of get rid of some of these columns. There's a column here, a name column. NA means there's nothing there of use, so let's get rid of that one. This ID column, we don't really need that, so let's get rid of that. And then this is just a little bit of example, just kind of getting rid of some other columns that, you know, I'm not sure they're going to be that useful, so let's just kind of get rid of those as well. So now when I run the df command, you can see that the file is smaller, right? It's still 569 columns. This is the number of actual samples, so the actual slides that you're looking at the microscope, but it's only 26 columns, so it's still a lot, but it's less than it was before. We're actually going to tune that down a bit more further. The thing you want to know is there any data that you need to fix? Means, are there any missing values? So if you run this piece of code, you can find out pretty quickly. This all looks like it's false. I haven't looked at everything, but you can actually use this command to see that it's all zeros. So there are no missing values, which is great. You can use the shape command to find out what the shape is of this matrix. We already kind of know it's 569 by 26, but this is another way to do it, 569 by 26. So the next step is now preparing our data. Now, the important part of this preparation is that we're going to do machine learning, and really the machine just wants numbers to go in, and then it's going to spit a number out. And it really doesn't care what those numbers are, but I'll explain a little bit more what that means. But let me go ahead and do this. We're going to map all the m's to 1 and the b's to 0. So now when we look at this and we run it, as you can see from before, these were all m and b, whereas now it's just 1s and 0s. Okay? More of something that a computer can read. Now, in order to create a machine learning algorithm, you need to create a training set and a test set. Fortunately, we can actually do that automatically by using scikit-learn. And scikit-learn allows us to basically split our data. So let's run this. And then let's run this. And let's also get a training set and a test set. There's going to be two basically matrices. One is going to be the training matrix that's going to train our model, give us all the weights, and then we're going to test it, and it'll be separate. And it's going to set the size of the test size to be 0.2 or maybe 20% of the total. We can now have the shape of these things. So these are two things. We have a training, which is going to have more for training, and then testing. There'll actually be 114 slides for testing and 455 slides for test. So that's actually pretty good. That's approximately 20%. The next step, as we kind of mentioned before, is we need to really normalize the data. The computer is not going to work. If some of the features are really large compared to smaller features, it's going to tend to use those larger features. So we want to normalize everything. So we can use that nicely using this function called min-max scalar. And there are lots of other functions there that make life a lot easier. So if you run min-max scalar on the training data set, you're going to get we're going to have called the same name. Now when we look at the new trained data set, it's a lot nice. Look at the means now. Here the diagnosis doesn't really matter, but the radius mean is 0.33, 0.3, 0.3, 0.2, 0.3. These are all numbers between 0 and 1. And so now all these are scaled roughly the same, so that way a certain feature doesn't give a lot more weight than some other feature. That is actually, an assumption that we're making. There may be some features we think we'd want to have more, but that's really not what we want to do. And put in the machine learning program the features equally weighted, and then the training will actually allow you to figure out which features are more important and which aren't. This is the test data set, which we also need to scale because we'll be testing it as well later. But we want to keep them very separate. So this is now your test data set.